Okay, we're going to convert these reduced cofactors to ATPs. And in order to do that, let's remind ourselves of the electron transport chain and the different complexes. And we can go from there. I'm not putting complex 2 on here because it doesn't pump protons, but let's think about those protons, right? Complex 1, for every 2 electrons, right, pumps 4 protons. Complex 3, for every 2 electrons, can pump 4 protons, and complex 4, for every 2 electrons, pumps 2 protons. Okay, so where are these reduced cofactors entering into the electron transport chain? Well, let's do a different color here. Right, all of our mitochondrial NADHs start by dumping those 2 electrons on complex 1. So for each of these NADHs, we can think of them as pumping a total of 10 protons across. 4, 4, 2. Okay. Our cytoplasmic NADHs end up pumping protons in between complex 1 and 3. Actually, at complex 2b, a mitochondrial dehydrogenase accepts those cytoplasmic electrons, converts them eventually to QH2. And so we can think of those cytoplasmic NADHs as only pumping across 6 protons from complex 3 and complex 4. Our FADH2 also enters into the electron transport chain as a way of making QH2, right? So it can only pump 6 protons across. Okay, now pumping the protons across doesn't directly make ATPs, but it's this proton gradient which drives, right, our ATPase at complex 5, which really pumps, you can think of it as a proton pump also, pumps those protons back across the intermembrane or inner membrane from the intermembrane space. Maybe we should label that, right? This is the intermembrane space, and this is the matrix. So the ATPase really relieves this gradient, right? Brings it back to an equilibrium by pumping the excess protons that are in the intermembrane space back over to the mitochondria, and in that process generates ATPs, right? And so what is the accounting here? How many protons do we need to pump across the ATPase in order to make an ATP? Well, it differs between different species. It really differs depending on how many C subunits make up a rotor of this ATPase. But for our accounting purposes, we're going to say that it takes about 10 protons to turn this ATPase one complete rotation. And one rotation generates 3 ATPs because there are 3 separate catalytic subunits in an ATPase, and if we rotate it around once, each of those catalytic subunits has the ability to make an ATP. Okay, so what does that mean in terms of our accounting here? Well, for our NADHs that are mitochondrial, they can turn the ATPase one complete turn to give 3 ATPs. So this 2 NADHs will give us 6 ATPs. 3 for each NADH. Here, 18 ATPs. Okay, our GTPs we count just like an ATP. Our cytoplasmic NADHs can really only turn this about 2/3 of the way, so we're going to convert that over roughly to 2 ATPs each, right? So instead of turning the ATPase a complete revolution, we're doing about 2/3 of that, so we can get about 2 ATPs. So these two NADH stars, 4 ATPs. These two FADH2s also give us about 4 ATPs. Okay, so let's total this up, right? This is from one glucose molecule. We get 2 plus 4 is 6, 12, 30, 32, 36. Okay, and this is a number that you may have heard about, and it varies, right? We had to do some assumptions in here, but roughly, you know, one glucose molecule completely combusted in the presence of oxygen, going through glycolysis and the Krebs cycle. This 6 carbons of this glucose, you know, are burned off with 6 carbon dioxides and result in enough energy to give us 36 ATPs. Okay. We can also have accounting here that relates, you know, these reduced cofactors to number of protons too, and that can be important when we have inhibitors that block one of these complexes. You know, what is the effect on ATP production? Well, it depends on how many protons can be pumped across in the presence of these inhibitors, and we'll see some examples of that in class.